He wailed the blues at night. He wailed, he wailed, he wailed just right.
Conventional science recognizes that all matter, each molecule of everything on Earth and beyond, oscillates at its own distinct frequency.
Can the amplified voice of Ella Fitzgerald shatter this glass?
The atoms that come together to form a molecule are held together with an energy bond that both emits and absorbs its own specific electromagnetic frequency. No two species of molecules have the same electromagnetic oscillations or energetic signature. By increasing the intensity of the harmonic frequency of the molecular structure of the wine glass, science can cause the glass to shatter.
Believe it.
This is a commonly accepted law of nature, so why can't other molecules be isolated and destroyed in the same way? Why can't the oscillation frequencies of killer viruses like cancer and AIDS be identified and destroyed? Why hasn't someone performed the research? Well, someone has. Royal Raymond Rife was a brilliant scientist who was born in 1888 and died in 1971. His studies at Johns Hopkins University led to his development of the technologies that are still commonly used today in the fields of optics, electronics, radiochemistry, biochemistry, ballistics, and aviation. He received 14 major awards and honors, including an honorary doctorate from the University of Heidelberg. During the 30 years that Rife spent designing and building medical instruments, he worked for Zeiss Optics, the U.S. government, and several private benefactors. Because Rife was self-educated in so many different fields, he intuitively looked for his answers in areas beyond the rigid scientific structure of his day. He had mastered so many different disciplines that he literally had at his intellectual disposal the skills and knowledge of an entire team of scientists and technicians from a number of different scientific fields. So whenever new technology was needed to perform a new task, Rife simply invented and then built it himself. In 1920, Rife began investigating the possibilities of treating disease with electricity. He discovered that each disease he studied had different electrical characteristics and started subjecting these organisms to different electrical frequencies. His first virus microscope complete, Rife performed tens of thousands of lab tests in an effort to isolate the microorganism that caused tuberculosis. Traditional scientific procedure requires the staining of samples to make them visible under the microscope. Unfortunately, the minuteness of the viruses made them impossible to stain with the existing acid-based stains. Rife conceived a method of staining the samples with light and began building a microscope that would enable the frequency of light to correspond with the electrical frequency of the microorganism under observation. His cancer research began in 1922, but it would take until 1932, after thousands of tests, that he was able to isolate the microorganism which he named the BX virus. By 1933, he had perfected that technology and had constructed the incredibly complex universal microscope, which is capable of magnifying objects 60,000 times their normal size. With this incredible microscope, Rife became the first human being to actually see a live virus. Until quite recently, the universal microscope was the only microscope able to view a live virus. Rife painstakingly identified the individual spectroscopic signature of each microbe. Colleagues of Rife reported his incredible patience, sitting at the microscope many times for over 24 hours straight without a break. Once he discovered the oscillation rate of a particular organism, Rife concentrated on refining his method of destroying it. He used the same principle to kill the virus as he had used to make the organism visible: light frequency resonance. By increasing the intensity of a frequency that resonated naturally within these microbes, Rife increased their natural oscillations until they distorted and disintegrated from structural stresses without harming the surrounding tissue. Rife called this frequency the mortal oscillatory rate, or MOR. Half a century later, doctors began using a like technology known as lithotripsy, where kidney stones are destroyed with high-energy shock waves. identify the MOR of cancer cells, turn up the volume and destroy them without damaging the host organism. It sounds like a scientific miracle, but will it work on the human body?
In 1934, a clinical study was set up at the Scripps Annex. Now, this is the Scripps Ranch. It was owned by Ellen Scripp. And there they set up a clinical study under the auspices of Dr. Milbank Johnson. And they had a team of physicians that examined terminal cancer patients. Every one of those patients was declared terminal by their group of scientists that was involved in this study. Dr. Rife would take some of the blood, examine, find the particular frequency for that organism for that patient, tune that instrument to the patient. Within 2 months, 14 of the 16 patients were declared cured by this team of physicians. It took another 6 weeks to cure the remaining 2, 100% cure rate.